Micturition is the act of urinating and is partly controlled by the spinal micturition reflex. When your bladder becomes approximately 200 milliliters or more full with urine, the stretch receptors inside the bladder wall become excited and issue afferent signals in the pelvic nerve by way of sensory fibers. These signals, here in green, head to the sacral spinal cord, usually in segments S2 and S3. Now, efferent signals from the spinal cord head back down to the bladder and to the parasympathetic ganglion in the bladder wall through motor fibers in the pelvic nerve. Now, this ganglion excites the detressor muscle, which relaxes the internal urethral sphincter, allowing you to urinate. This process is called the involuntary micturition reflex and is generally seen in young children and people with spinal cord injuries. Now, most of us have voluntary control of the urination process. Voluntary control happens when the stretch receptors sends a signal to the nucleus within the pons, so way up here in our brain, called the micturition center. If you are in a position to urinate, the pons will descend efferent signals down the sacral spinal cord, exciting the parasympathetic ganglion neurons, actually causing the stimulation and contraction of the detressor muscle, which relaxes that internal urethral sphincter, allowing you to urinate. But if you're stuck holding it because you are not near a restroom, signals from the cerebrum descend to all the way down to the external urethral sphincter, telling it to relax and close, preventing urine from escaping the urethra.